now that you've got all of your data downloaded onto your computer, there's no need to use anything else other than the script for doing most of the processing. We will go back to use a website called David later, but apart from that, everything can be done from within the workflow. So you've got your data in the files here, but what you actually need to do is get it inside R. So that's what this next command does. So what it does is it takes all of the different files, brings them all together and imports them inside R. So when it's finished, you'll see a new thing appear within inside R, which will be a very big set of data. Because what this is a big matrix. So you have one row for each of your patients, and you have a column for each one of the genes. So as well as that, doing that, creating the large element, it also saves them as a file so that you can import it later. So there's no need for you to go and get the data from the database ever again. You can just import these RDA, these are data files. So that's done it for both sets of data. One of them was quite slow because it's a big data file, the other one's faster. Next thing you want to do is remove any outliers. Now I set this to 0.6, actually it's better to set it to 0.8. So what you're doing is you're looking at the data and you're looking to see if any of the individual patients is very different to any of the others. So you're doing this by a correlation analysis. So you're looking at a correlation of 0.8. So I run that one, correlation of 0.8. So it detected that there's one outlier. So what you can do is you can look at this by looking at the script. So you can see here, well actually to be quite honest, I might fancy excluding a few more than one. The darker the colour, the closer they are to being identical to one another. When you compare a sample to itself, then you get this perfect correlation of 1, which is this red diagonal. These ones which are lighter colour are indicating that these ones are quite different to the others. And if you look here at the spread of the expression, there's about 5 or 6 samples that are very different to the others. So I'm going to actually adjust that, because looking at it, I think I possibly want it to be 0.85. So I'm going to adjust this to be 0.85 to exclude the data that I think is a bit questionable. So I'm going to, if I change it to 0.85 for one, I should change it for both sets of data. So this is first for the stage 1. So now I've removed 5, and this is stage 2, and I've removed none. So if I look at the stage 2 data, you can see that they're all very similar to one another. There's none that aren't drastically different. There's a lot of bouncing up and down in differences in the strength of the signal for the different data sets, but that will be coped with a process called normalization, which is the thing that happens next. So what you do is run normalization, and what that does is it makes sure that all of the samples end up with the same baseline or at the same intensity. Because if you're comparing how much of something there is between two different samples and they're at different intensities, then the most intense one, there's always going to appear to be more, even if there isn't. So the first thing you need to do is get the baselines sorted. So this will take the two separate data sets, put them together and on a single standard. that's all done. The next step does a filtering to get rid of anything that's particularly boring, because they're unimportant genes. So if you look here, after normalization, you've got a file which was 60.7 megabytes. Now you've got one which is 57.5, because quite a lot of the genes are not different between any of them, between any column and between any patient. They might be always 0 or always exactly the same number. Most likely always 0. Then I want to save that data set, so in case I do have a problem, I've got both of them combined together in this big single set of data. Next thing I want to do is add on labels to define which is group 1 and which is group 2, which are based on the bar codes which I defined earlier when I downloaded the data. And the last thing I want to do here is the actual differential gene expression. So the next line here, this diff expressed genes, is the thing that actually does the calculation of the differentially expressed genes. Comparing 179 samples from group 1 to 20 samples in group 2, which are across 34,561 features, which originally started with over 60,000. So we've reduced quite a lot in terms of uninteresting features. And the final thing I'm going to do is check how many genes it's come up with as being differentially expressed by saying how many rows are there in this variable called differentially expressed genes. And it's come up that there are 528. That's not bad. It's not massive amounts. It's not many thousands, which is not ideal because then it's difficult to interpret what it means. And it's not very few either. So it seems there are about 500 or 600 genes which are different between stage 1, small scale cancer, localized cancer, and metastasis.